Dear viewers, this video is part 1 in the 3-part series on cancer genetics in the Medicine for Anyone YouTube channel. Cancer is a genetic disease. Cancer arises through a series of somatic alterations in DNA that result in unrestrained cellular proliferation. Note, a somatic cell is any biological cell forming the body of a multicellular organism other than a gamete, germ cell or gametocyte, ovum or sperm. Such cells make the tissues of the body. Most of these alterations involve subtle sequence changes in DNA, that is, mutations. The somatic mutations may originate as a consequence of random replication errors or exposure to carcinogens, example radiation, and can be exacerbated by faulty DNA repair processes. Most cancers arise sporadically. Much less commonly, clustering of cancers occurs in families that carry a germline mutation in a cancer gene. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. The clonal origin and multi-step nature of cancer. Nearly all cancers originate from a single cell. This clonal origin is a critical discriminating feature between neoplasia and hyperplasia. Multiple cumulative mutational events are invariably required for the progression of a tumor from normal to fully malignant phenotype. The process can be seen as Darwinian microevolution, in which, at each successive step, the mutated cells gain a growth advantage, resulting in the expansion of a neoplastic clone. Note, cancer development within an individual is also an evolutionary process which, in many respects, mirrors species evolution. Species evolve by mutation and selection acting on individuals in a population. Tumors evolve by mutation and selection acting on cells in a tissue. Just 3 causal mutations are required for the development of several of the most common cancers. Overall, it is currently believed that most common solid tumors require a minimum of 3 mutated cancer driver genes, either oncogenes or tumor suppressor genes, for their development. 1 or 2 mutations are sufficient for benign tumorigenesis, but not for the invasive capacity that distinguishes cancers from benign tumors. Less common tumors, such as liquid tumors like leukemias or lymphomas, sarcomas, and childhood tumors, appear to require only 2 driver gene alterations for malignancy. Mutations required from a normal cell DNA structure for benign tumors 2, leukemias or lymphomas 2, childhood tumors 2, sarcomas 2, other malignancies 3. Note that a cancer driver gene is best defined as one containing a mutation that increases the selective growth advantage of the cell containing it. Normally, cell birth and cell death are in perfect equilibrium. Every time a cell is born, another in the same lineage dies. Cancer driver gene mutations alter this equilibrium so that more cells are born than die. The imbalance between normal cells and cancer cells is often slight, so that the difference between cell birth and cell death can be less than 1%. This explains, in combination with the low rate of mutation, why tumorigenesis, the journey from a normal cell to a typical malignant solid tumor, often takes decades. We now know the precise nature of the genetic alterations responsible for nearly all malignancies and are beginning to understand how these alterations promote the distinct stages of tumor growth. This figure shows the different stepwise progression of colon carcinoma. Carcinogen-induced mutations initially occur, then additional driver mutations occur. Later, additional mutations with emergence of subclones aiding invasion and metastasis occur. The prototypical example is colon cancer, in which analysis of genomes from the entire spectrum of neoplastic growths from normal colon epithelium through adenoma to carcinoma have identified mutations that are highly characteristic of each type of lesion. See figure shown earlier, that is progressive somatic mutational steps in the development of colon carcinoma. The accumulation of alterations in a number of different genes results in the progression from normal epithelium through adenoma to full-blown carcinoma. Genetic instability, microsatellite or chromosomal instability, accelerates the progression by increasing the likelihood of mutation at each step. Patients with familial polyposis are already one step into this pathway because they inherit a germline alteration of the APC gene. Explanatory note. Microsatellite, that means MIN. These are regions of repeated DNA that change in length, showing instability when mismatch repair is not working properly. Chromosomal instability, that is SIN. This results from errors in chromosome segregation during mitosis, leading to structural and numerical chromosomal abnormalities. APC protein in chromosome 5q22. The APC protein acts as a tumor suppressor, which means that it keeps cells from growing and dividing too fast or in an uncontrolled way. It helps control how often a cell divides, how it attaches to other cells within a tissue, and whether a cell moves within or away from a tissue. Beta-catenin in chromosome 3p22. Beta-catenin acts as a transcriptional activator, regulates transcription of target genes responsible for cell proliferation and differentiation. Beta-catenin is the key effector responsible for transduction of the signal to the nucleus, and it triggers transcription of certain genes responsible for the control of cell fate decisions in many cells and tissues. KRAS gene in chromosome 12p12. KRAS gene is a gene that makes a protein, KRAS, that is involved in cell signaling pathways that control cell growth, cell maturation, and cell death. The natural unchanged form of the gene is called wild-type KRAS. BRAF gene in chromosome 7q34. BRAF is a gene found on chromosome 7 that encodes a protein also called BRAF. This protein plays a role in cell growth by sending signals inside the cell. SMAD4 protein in chromosome 18q21. The SMAD4 protein serves both as a transcription factor and as a tumor suppressor. The TGFB2 gene in chromosome 1q41. The TGFB2 gene provides instructions for producing a protein called transforming growth factor beta 2. This gene encodes a secreted ligand of the TGF-beta superfamily of proteins. They regulate gene expression. TP53 gene in chromosome 17p13. The TP53 gene provides instructions for making a protein called tumor protein p53. This protein acts as a tumor suppressor. Chromosomal abnormalities associated with some common cancers. Breast cancer. The breast cancer susceptibility gene BRCA1 is located on chromosome 17q 12 to 21. That is chromosome 17q, that means long arm, region 12 to 21. BRCA1 encodes a tumor suppressor protein that acts as an inhibitor of tumor growth and is involved in DNA damage repair. Mutation of BRCA1 increases the risk of developing breast cancer. What are BRCA1 and BRCA2? BRCA1 breast cancer gene 1 and BRCA2 breast cancer gene 2 located in 13q12 to 23 are genes which produce proteins that help repair damaged DNA. They are also called tumor suppressor genes. Everyone has two copies of each of these genes, one copy inherited from each parent. People who inherit harmful variants in one of these genes have increased risks of several cancers, most notably breast and ovarian cancer. A harmful variant in BRCA1 or BRCA2 can be inherited from either parent. Each child of a parent who carries any mutation in one of these genes has a 50% chance or 1 in 2 chance of inheriting the mutation. Even if someone has inherited a harmful variant in BRCA1 or BRCA2 from one parent, they would have inherited a normal copy of that gene from the other parent. But the normal copy can be lost or changed in some cells in the body during that person's lifetime. Such a change is called a somatic alteration. Cells that don't have any functioning BRCA1 or BRCA2 proteins can grow out of control and become cancer. Lung cancer. Somatic mutations in the TP53, that is chromosome 17p, EGFR in chromosome 7p11, and CRAS, chromosome 12p 12.1 genes are common among lung cancers. The TP53, see also Cancer Cell Biology, Medicine for Common Man series. This gene provides instructions for making a protein called P53 that is located in the nucleus of cells throughout the body, where it attaches, that is binds directly to DNA. The protein regulates cell growth and division by monitoring DNA damage. When DNA becomes damaged, P53 helps determine whether the DNA will be repaired or the cell itself will self-destruct, that is undergo apoptosis. TP53 gene mutations result in the production of an altered P53 protein that cannot bind to DNA. The altered protein cannot regulate cell proliferation effectively and allows DNA damage to accumulate in cells. Such cells may continue to divide in an uncontrolled way, leading to tumor growth. Mutations in the EGFR or CRAS gene lead to the production of a protein that is constantly turned on, that is activated. As a result, cells constantly receive signals to proliferate, leading to tumor formation. When these genetic changes occur in the cells in the lungs, lung cancer develops. The EGFR and CRAS genes each provide instructions for making a protein that is embedded within the cell membrane. The EGFR gene provides instructions for making a receptor protein called the epidermal growth factor receptor, which spans the cell membrane so that one end of the protein remains inside the cell and the other end projects from the outer surface of the cell. This positioning allows the receptor to attach or bind to other proteins called ligands outside the cell and to receive signals that help the cell respond to its environment. Ligands and receptors fit together like keys into locks. Epidermal growth factor receptor binds to at least 7 different ligands. The binding of a ligand to epidermal growth factor receptor allows the receptor to activate the receptor complex. As a result, signaling pathways within the cell are triggered that promote cell growth and division, that is proliferation and cell survival. CRAS gene, also known as CRAS proto-oncogene, GTPase, that is guanosine triphosphatase. The CRAS gene provides instructions for making a protein called KRAS, that is part of a signaling pathway known as the RAS-MAPK pathway. The protein relays signals from outside the cell to the cell's nucleus. These signals instruct the cell to grow and divide, that is proliferate, or to mature and take on specialized functions, that is differentiate. The KRAS protein is a GTPase, which means it converts a molecule called GTP, that is guanosine triphosphate, into another molecule called GDP, that is guanosine diphosphate. In this way, the KRAS protein acts like a switch that is turned on and off by the GTP and GDP molecules. To transmit signals, it must be turned on by attaching or binding to a molecule of GTP. The KRAS protein is turned off, that is inactivated, when it converts the GTP to GDP. When the protein is bound to GDP, it does not relay signals to the cell's nucleus. The KRAS gene belongs to a class of genes known as oncogenes. When mutated, oncogenes have the potential to cause normal cells to become cancerous. The CRAS gene is in the RAS family of oncogenes, which play important roles in cell division, cell differentiation, and the self-destruction of cells, that is apoptosis. GTP. GTP is an energy-rich nucleotide analogous to ATP, that is composed of guanine linked to ribose and 3 phosphate groups, and is necessary for peptide bond formation during protein synthesis, also called guanosine triphosphate. Guanosine, a nucleotide, is composed of a guanine, that is a nucleobase, and a ribose ring. It plays a vital role in the synthesis of nucleic acids and proteins. Melanomas, alteration in BRAF gene, which is in chromosome 7q34. BRAF is a serine threonine protein kinase activating the MAP kinase ERK signaling pathway. Mitogen activated protein kinase MAPK cascades are key signaling pathways that regulate a wide variety of cellular processes, including proliferation, differentiation, and apoptosis. So, dear viewers, this is the end of part 1 in the 3-part series on cancer genetics in the Medicine for Anyone YouTube channel. Thank you for watching the video.